Good afternoon, genetics students. Epigenetics can be categorized in the time scale involved in what's known as epigenetic memory. What that means is how long is the modification going to be around for in an individual cell or an organism. The first category is cellular memory. Mitotically heritable transcriptional states are established during development, and these states are carried on into subsequent cells that arise from that given cellular state. The best example of this is that patterns are maintained. So, for example, cellular memory includes things like heart cells only expressing the genes that are required for heart cells to function, and liver cells only expressing the genes that are required for liver cells to function. This cellular memory is a situation, again, that arises because all of the genetic information is present in every single cell. However, not all of the genes are needed to be transcribed in every single cell. So these modifications that occur very early on in development in response to developmental cues are maintained in the cellular components that arise from that point. Transcriptional memory is on a much smaller time scale with respect to how long the modifications are around and continue to play a role in the way transcription occurs. These are typically environmental cues that occur and allow for quick response to take place. Components needed for modification stay associated with DNA and proteins for a shorter period of time, again, typically in quick response to environmental cues. So transcriptional memory is typically much shorter than cellular memory overall. And lastly, what occurs is known as transgenerational memory. This is a type of epigenetic memory where changes or modifications are actually passed on to the next generation, and we will see a classic example of that in the next series of slides. The scientists in this study took this one step further and analyzed the pups under the low stress condition compared to the pups that were under high stress condition. And again, low stress was due to mothers that were being attentive. High stress circumstances or situations were mothers that were unattentive or inattentive to their pups. It turns out that these modifications that were identified in the two situations were maintained into the next generation. Interestingly, these scientists conducted another set of experiments where they switched the pups at birth. So pups that were born to attentive mothers were switched with pups that were born to inattentive mothers. Again, the pups were switched from the type of mother they were born from. The brains from these mice were also analyzed, and the same circumstances were found. Those pups that were raised by inattentive mothers had higher stress components and a higher number of genes that were modified epigenetically. There are different mechanisms that are associated with epigenetic modification. There is what's known as DNA methylation, which is a modification that occurs directly to the DNA. There is histone modification, which is a modification that occurs to the histone proteins that are in close association with the DNA in eukaryotic cells. And there is RNA silencing, where RNA is in fact silenced, meaning not allowed to be translated. So the first mechanism is known as DNA methylation. This involves a covalent addition of the methyl group to cytosine, and the methyl makes DNA more compact. This prevents transcription and inhibits gene expression. There are a whole family of enzymes that are referred to as DNA methyltransferases that play a role in this addition of methyl groups that then affects transcriptional patterns. The next category is known as histone modification. Histones are a family of proteins that are associated with DNA that allow for the complex organization and compaction of the genetic material in the nucleus of a eukaryotic cell. There are, in fact, multiple types of histone modifications that we know exist. Acetylation does play a role in loosening up the DNA, which could allow for a transcription pattern to take place. Methylation can also occur to histones. This can either loosen up the DNA or tighten up the DNA, depending upon where specifically the methyl group is added. Lastly, phosphorylation is a third type of histone modification. This occurs to serines and threonines in the histone proteins themselves, and this makes the DNA very, very compact, hence inhibiting transcription. And the last broad category is known as RNA silencing. Expression is downregulated by what are known as small non-coding RNAs. This represents about 1% of the genome and targets about 30% of genes by either blocking translation or involved in degradation of the message overall.